Namaskar friends, these visuals explain the debate over the spontaneous generation theory. During year 300 BC to 1684 AD, theory of spontaneous generation was hypothesized to answer the query how life is generated. This theory proposes that organisms arise from non-living matter. The foremost supporter of this theory was Aristotle. The follower of this theory suggested the various evidences in support of it. This includes mushrooms developed from the decaying wood, maggots generated from the meat, scorpions generated from the cow dung, frogs generated from the mud, and mice generated from the decaying wheat bran or old rags. There is nothing incorrect about the observation themselves. It is a conclusion drawn from them were not based on the experimental evidences. This theory is also known as abiogenesis. The spontaneous generation theory was disproved experimentally by various scientists like Francisco Redi, Louis Joblet, Lazzaro Spallanzani, Franz Schulze, Theodor Schwann, Schroeder and Deutsch, Louis Pasteur, and John Tyndall. Friends, all these experiments are conducted at a different nation and during the period of nearly 200 years. Francisco Redi made a first attempt to disprove the theory of spontaneous generation. He placed meat in two jars. One jar was covered with a fine gauze while other kept open. The flies were developed only in open jar and not in a closed jar. From experiment, it is concluded that maggots were developed from the maggots but not from the non-living meat. Similar experiment was performed by the Louis Joblet. He divided boiled meat infusion into two heat-treated flasks. One was open and other was closed to air. The microbial growth was appeared only in open flask, while no growth was detected in closed flask. Lazzaro Spallanzani performed same experiment by taking properly boiled meat in the airtight sealed flask. Also, as a control, a flask kept open to air. After several days, he found the control flask with a swarming of microorganisms, but a sealed flask contains no microorganism. Meanwhile, in 1775, Priestley, Cavendish, and Lavoisier discovered the oxygen and its essentiality for the life. By considering this fact, air shoes pass the air through the sulfuric acid and potassium hydroxide solution to previously heated infusion of meat, while T-Schwann passed the air through the hot glass tube to previously heated infusion of meat. The infusion exposed to such air remains organism free after several days. Schroeder and Deutsch performed similar experiments, but they have supplied the filtered air through the cotton in the infusion containing flask. This prevented the appearance of microorganisms in flask. This experiment was origin of use of cotton plugging for the microbial culture tubes. All these experiments were keenly studied by Louis Pasteur. He drawn an important conclusion that microbes are associated with the dust present in air, which is excluded by various ways in this experiment. In subsequent visuals, we will know how the theory of abiogenesis was disproved finally. Thank you for listening me patiently.